There have been a number of recent developments in the field of lipid lowering and cardiovascular disease that may actually affect how we actually begin to look at PCSK9 inhibitors and their role in the management of people with atherosclerosis. For many years we have thought that the process of atherosclerosis involves an interaction between cardiovascular risk factors, and at the level of cell biology and the arterial wall there is well known to be an interaction between cholesterol molecules, the immune system and inflammation. And one of the great questions initially raised by the statins was their anti-inflammatory effect part of their action? Now we actually have further data. We've known for a little while that in the fields of autoimmune disease that use of the TNF inhibitors or any agent that basically damped disease reduced cardiovascular events in those groups. But now we have a specific drug targeting the IL-1 beta receptor, canakinumab, which has been investigated in a phase 3 study. This IL-1 beta receptor antagonist actually reduces the rate of cardiovascular events in that population and has no effect whatsoever on lipids. So that means that we have a primary drug targeting atherosclerosis and one of the principal forms and methods of immune activation in atherosclerosis, and showing a reduction, at least preliminary, in events. I think this is very, very interesting because we've known about this interaction around immune disease and atherosclerosis for a while, and it means that we might be able to start thinking that if the endpoints trial with canakinumab is as promising as the initial data, that we may need to manage and subgroup some of our patients with atherosclerotic disease. So thus those that have autoimmune diseases such as rheumatoid or lupus might actually benefit more from being on an immune system agent like canakinumab than they would benefit from being on additional lipid lowering therapy beyond the standards that we would normally prescribe. The other interesting area has been really the renaissance of a class of compounds that have been in disgrace for a period of 3 or 4 years. For many years we have known that there is an interplay between HDL and LDL, which is mediated through the cholesterol transport protein, or CETP. This led to the development of a whole class of compounds called CETP inhibitors, but the results with them have to date been very, very disappointing. The first drug in the series, torcetrapib, bound covalently onto HDL particles, also activated systems raising blood pressure and causing arrhythmias, and had to be withdrawn. The second drug in the series, dalcetrapib, was a far weaker agent, deliberately designed so it didn't change LDL cholesterol very much, but did raise HDL by about 30%. This was tried in an acute coronary syndrome trial and unfortunately had no effects. The third agent was actually going back to the lipid efficacy of torcetrapib. It was called evacetrapib, and it raised HDL by about 110 to 130%, reduced LDL cholesterol by about 30% to 40%. And this was again tried in an acute coronary syndrome population, but the trial was abandoned after about 4 years for futility. That's where the field stood really until this week. We have had a fourth agent around. This is a drug called anacetrapib. It raises HDL by about 110%, almost doubling it. It reduces LDL cholesterol by, again, 35 to 40%. No blood pressure effects, no adverse events that could be seen. It went into a large trial of 25,000 patients on top of statin therapy that had been optimized. This is the REVEAL study, and the headline results have been announced this week. This shows that the drug has made its primary efficacy endpoint of reducing combined cardiovascular events, fatal and non-fatal, after 5 years of treatment. The design paper implies that this means that it must have reduced cardiovascular events by about 10%. It is also interesting to note that the patients going into this study had an LDL cholesterol on optimal treatment of 1.5 millimoles per litre. In FOURIER, with a PCSK9, it was 2.3 millimoles per litre. In the on-treatment arm in IMPROVE-IT, it was 1.4 millimoles per litre. So these people are starting where patients stopped from IMPROVE-IT. They're a slightly different group. They're not acute coronary syndrome, they're chronic cardiovascular disease. But nevertheless, it shows that going lower, we still get an event benefit. And if you run an approximate calculation based on the CTT analysis, which holds for PCSK9 inhibitors, holds for ezetimibe, and was originally derived from statins, the 10% reduction is roughly what you'd expect for using this type of drug based on LDL reduction alone. So it seems that it's going to be all to play for in the lipid lowering game.